Hi, and thank you for listening to our presentation on Metabarcoding Saproxylic fungi communities in Franklin Parker Preserve, New Jersey. This research was conducted by a group of undergraduate students from Kean University during the spring 2020 semester. I'll be your host, Cade Barisha. Now without further ado, let's begin. What are fungi? Fungi consist of their own kingdom. They are typically multicellular, aerobic eukaryotes. Most fungi are filamentous, contain chitinous cell walls, and lack flagella. Forest microbiomes consist of multiple fungal species, which inhabit the soil, plants, and wood. Our research revolves around the decomposition process in the forest, with major decomposers being fungi. What are saproxylic fungi? Saproxylic fungi are fungal species within a forest microbiome that inhabit dead wood. A forest microbiome consists of multiple fungal species which inhabit soil, plants, and wood. Microbiomes are mostly responsible for decomposition, supporting normal functionings of the forest ecosystem. The image on the slide is exactly what we're looking for, a well-rotted piece of log inhabited by multiple unknown fungal species. Why are saproxylic fungi important? They are the only organisms that can digest lignin, a tough woody polymer, leading to the recycling of nutrients, soil formation, erosion protection, as well as the release of carbon into the atmosphere. Why do we need to know about saproxylic fungi? Well, saproxylic fungi live in guilds that interchange each other with the progression of wood decay. More aggressive fungi digest first, then next guilds arrive and continue to digest softened material. If some species are missing, the complete decay might be delayed for a long period of time, which is what we want to avoid. Also, many insects, young plants, and animals, such as woodpeckers, depend on the decayed wood as a habitat. Do saproxylic fungi require conservation? Diversity of saproxylic fungi might be affected by park management techniques, such as collecting or burning wood. In a forest, logging can also affect the overall amount of substrate required for fungi to live in. There are many ways to throw off the balance in a microbiome. Therefore, the work we do is paramount in providing data for the effects of saproxylic fungi. The extent of human impact on diversity of saproxylic fungi in the US is still under research. This is because most of the saproxylic fungi are cryptic, hard to recognize, or microscopic, so it is not easy to find nor identify them. Our objective is to use DNA metabarcoding method to recover complete information of microbiomes from the wood logs and standing dead wood from Franklin Parker Reserve, New Jersey. This information is to comprise a list of saproxylic fungi living in this area so it can be further compared to the areas with an intense human impact. To do so, the results will be published in the Global Biodiversity Information Facility (GBIF) database. The identification of saproxylic microbiome. Due to the nature of saproxylic fungi, dead wood would need to be drilled and the remaining sawdust will have to be collected and brought to the lab for DNA isolation. Here we have a video of one of the students performing the collection process. Once collected, we identify saproxylic fungi using metabarcoding. It was brought to the lab for DNA isolation and purification. Methods and procedures. Once the sawdust was crushed, preparations for metabarcoding and PCR had to be done. Each of the 6 sawdust samples from each individual tree were subjected to PCR with the same pair of primers, but different tags, in order to differentiate them. The table you see on the slide are each of the sequences used for amplification by PCR, with their corresponding primer name. ITS barcoding gene in fungi. The ITS regions of nuclear ribosomal DNA are the selected DNA regions for fungal species identification. Fungal DNA have internal transcribed spacer regions, ITSs for short, that assist in DNA barcoding. 6 DNA samples were subjected to PCR using tagged primers. The primers used in PCR are used to amplify the ITS regions. For each sample, the same primer was used, that being ITS7 as forward and ITS4 as reverse. Continuing on with methods and procedures, the PCR products were then visualized and confirmed by gel electrophoresis. 4 of the 6 generated DNA fragments were around 300 to 400 BP. The gel images, unfortunately, were left in the lab and cannot be accessed due to the COVID-related lockdown at the Kane University campus. Following the gel electrophoresis, the PCR products were then purified using magnetic beads, and the concentration of the purified samples were measured using a Quippet fluorometer in order to ascertain the quality of the sample. The way the Quippet fluorometer does this is that it detects fluorescent dyes that are specific to the target of interest. These fluorescent dyes emit only when bound to the target molecules, helping determine the concentration of either nucleic acids or proteins. The purified DNA samples were then pooled together in equal concentrations and the multiplex sample sent to Genoese, New Jersey for next generation sequencing using Illumina 2x250 technology. The SCADA pipeline was then used, and in order to demonstrate this process more clearly, on the screen will be an accompanying video summary. Once logged in, we first upload the file we've obtained from Genoese. Those very files contain the previously mentioned PCR fragments. Then we upload a primer and tag set, which allows the software to sort the files by grouping the data into clusters. This information will then be conformed into a dataset by feeding into this bioinformatic pipeline the information we've gathered so far. This pipeline will not just organize the data, but also remove any data that are low quality, such as a sequence being incomplete. Due to this, the majority of the sequences were removed due to low quality. Once that process is done, the data will then be placed under a specific set of parameters in order to cluster the group of sequences that belong to specific species and specific tags. Once said parameters have been set, we go to the Jobs tab to submit the job, where it will be compared to an ITS database which contains sequences of fungi. The submissions will contain some species that are unable to be identified due to certain sequences not meeting our set parameters, at which case we then input the unidentified sequences manually through BLAST. That has summarized our whole bioinformatic portion of the presentation. Now to the results. The table you see before you are the fungi in some host trees that were identified. The complete list of identified species was published as a dataset at the previously mentioned Global Biodiversity Information Facility, gbif.org. To do this, we use Darwin's Core, which is a standard in sharing information on biological diversity. This standard was used to format our data, which was then eventually uploaded, using the Integrated Publishing Toolkit. Datasets published at gbif.org are available for all biodiversity, ecology, and biology specialists from all over the world. You can find there the list of species, location, time of collection, and other important data for your use. The conclusion. The identified species were recorded to not just contribute to our knowledge on biodiversity of saproxylic fungal species, but to also help assess diversity for future natural conservation studies. Though the data collected throughout was quite limited, it is still a step in the right direction. It is apparent that the 6 tree samples used are not enough to completely ascertain the diversity found at Franklin Parker Preserve, and future sampling of the area would be required. Also, the results do not exclude any species samples whose results came out negative due to the high likelihood that some trees simply did not have any growth of fungi. This could be caused by a variety of reasons, such as the lack of valid transportation or some form of human obstruction. But I digress. To finish this off on a high note, the data recorded will make strides in future conservation as well as the mapping of saproxylic fungi. That will conclude our presentation, and I would like to thank you for listening because it was truly a pleasure. For any further information on the collection of resources, please check the reference slide. And with that, I hope you all have a wonderful day and stay healthy.